The 20th century has been a period of rapid advancements in the area of genetics. It wasn't until this century that scientists began to understand the chemical substance responsible for directing the development of an organism. This century-long investigation led to the discovery of the universal code of life, DNA. In this program, we will learn about important experiments that gave scientists insight on the chemical nature of DNA. We will also learn how DNA replicates itself to produce an exact copy of the original molecule. How did scientists find out that DNA is the chemical substance responsible for heredity? Many scientists worked very hard on discovering this substance, but it wasn't until 1928 that Frederick Griffith inadvertently uncovered a clue in the genetic mystery. He was attempting to develop a vaccine against pneumococcus, the bacterium that causes pneumonia. While doing this, he discovered that an unknown substance inside the bacterium could transform one type of bacterium into another if the two types were mixed together. Griffith worked with two strains of pneumococcus. The strain that caused the disease was called the smooth strain, because it appeared large and smooth. The second, which did not cause the disease, was called the rough strain, because it looked smaller and rougher than the smooth strain. Griffith's experiment involved infecting mice with both strains of the pneumococcus. As the diagram illustrates, he first infected mice with live rough type, and the mice survived. He then infected mice with a live strain of the smooth, disease-causing type, and the mice died. Next, he infected mice with heat-killed smooth type, and again the mice survived. The unexpected results occurred when he mixed heat-killed smooth type with live rough type. The expected result would be mice that lived. However, these mice also died. Griffith then looked for the bacteria in the dead mice and found living smooth type. The rough strain was harmless, and the smooth strain had been heat-killed. So what event must have occurred before the rough strain could become the disease-causing smooth strain? Griffith hypothesized that the material responsible for giving the smooth strain its lethal characteristics had transformed the rough type. This means that the genetic material passed from the smooth type into the rough type, transforming the rough strain into a smooth strain. This process is called transformation. Griffith did not know, however, what the genetic material was. The next famous experiment was performed in order to understand this mysterious substance better. Griffith's experiment was a good start at discovering the chemical responsible for directing a cell's activities and development. The next significant experiment was performed by Oswald T. Avery, Macleod, and McCarthy in 1944. This team of scientists set out to discover which component of the cell makes up the hereditary material. They took many pneumococcus cells and made extracts of the chemical compounds contained in the cells. They gradually removed one chemical compound after another, first the lipids, then the proteins, next RNA, and so on. In other words, they separated all of the cell's contents until they had pure samples of each chemical compound. Each time they tested to see if this was the material that passed into the rough strain, causing it to take on smooth strain characteristics. However, none of these components caused transformation. The last substance the researchers tested was a clear, stringy substance that could be wound around a glass stirring rod. When dissolved, this material was capable of transforming the rough strain into the disease-causing smooth strain. The team had discovered the hereditary material that caused the rough strain to take on smooth strain characteristics. Chemical analysis showed this chemical compound to be deoxyribonucleic acid, or DNA. Many people did not believe that DNA was the hereditary molecule because it did not seem complex enough to provide a blueprint for life. In the 1950s, Alfred Hershey and Martha Chase did an experiment that established beyond question that DNA is the hereditary material. They worked with an intestinal bacteria called E. coli and a virus that infects bacteria called a bacteriophage or simply phage. The researchers began the experiment knowing several things about the phages they were working with. They knew the shape of the phage and the mechanism by which the phage injected its contents into a bacterium. First, the phage attaches to the outside of the cell and injects its contents, leaving the phage body behind. Once this material is injected, new phage are produced until the cell bursts, releasing newly synthesized phage. Because new phage were made in the cell, the researchers knew that the hereditary material must pass into the cell. They also knew that a phage is made of only two things: DNA and protein. There are no other chemical components to a phage. Since the body remains outside of the cell after infection, the researchers assumed that the contents of the phage, which were injected into the bacterium, had to be the hereditary material. Their goal was to find out for certain whether this substance was protein or DNA. The researchers had a few chemical clues that helped them determine which compound was injected. First, DNA contains phosphorus, but protein does not. Second, protein contains sulfur, but DNA does not. Both phosphorus and sulfur can be made into radioactive isotopes, which means they emit radiation. This radiation then acts as a signal that scientists can measure experimentally. The research team grew phage-infected bacteria in a medium containing the radioactive phosphorus. Phage that replicated in this medium had incorporated radioactive phosphorus, thus producing hot DNA. They also grew phage-infected bacteria in a medium containing radioactive sulfur. Phage that replicated in this medium had incorporated the radioactive sulfur and thus contained hot protein. Next, Hershey and Chase infected bacteria that had grown in a medium containing no radioactive elements. One culture of cells was infected with the phage containing hot DNA, and another culture was infected with phage containing hot protein. After the phage injected their contents into the bacteria, the researchers separated the phage body from the cell surface with an electric blender. After separating the empty phage bodies from the infected cells, they studied the cells to see whether protein or DNA had been injected. They found the radioactive DNA inside the bacteria. They did not find radioactive protein in the bacteria. Therefore, the researchers concluded that DNA is the hereditary material. A scientist named P.A. Levine had already determined the chemical composition of DNA in the 1920s, but at that time, he did not know it was the hereditary material. He found that DNA is composed of three chemical components: a base, a sugar, called deoxyribose, and a phosphate group. There are four types of bases: adenine, guanine, cytosine, and thymine. One of these, in addition to deoxyribose, the sugar component of DNA, and a phosphate group, make up a nucleotide. Because there are four different bases, there are four different nucleotides. A DNA molecule consists of many nucleotides, some molecules being more than 1 million nucleotides long. After the chemical makeup of DNA was known, the next step was to find out the structure. What do you think a DNA molecule looks like? In 1953, James Watson and Francis Crick developed a model for DNA for which they later received the Nobel Prize. A third researcher, Rosalind Franklin, also made major contributions, but she died before she could receive the award. Watson and Crick, with the help of Franklin's X-ray studies of DNA crystals, determined that DNA is in the form of a helix. After trying many different arrangements, the researchers found two chains of sugar phosphate groups running parallel to each other, as shown in the diagram. This structure resembles a twisted ladder. The rungs of the ladder represent the base pairs that link the chains together. The twisting of the ladder forms the double helix. Each rung of the ladder is either an adenine paired to thymine or guanine paired to cytosine. Watson and Crick's model explained why one always finds the same amount of A and T and of C and G. The sequence of nucleotides varies greatly in a DNA molecule, but as the diagram illustrates, A always pairs with T and C always pairs with G. Knowing the structure of a double helix and the significance of the complementary pairing of bases led Watson and Crick to an important concept called replication. DNA replication is the mechanism whereby identical copies of a DNA molecule are created. A basic overview of replication begins with a DNA molecule, which consists of two strands of DNA that are paired with their complementary bases, of adenine to thymine, or guanine paired to cytosine. In the first step of replication, the double helix unzips, and the two strands separate. The separated strands become templates along which complementary nucleotides attach themselves to form new strands. Two copies identical to the one original DNA molecule are formed. Each new molecule consists of one strand from the original molecule, known as the parent strand, and one strand made from the free-floating bases, known as the daughter strand. This type of replication is called semi-conservative replication. Through experiments, researchers have determined that replication starting points occur at many places along the strand. Other biological molecules, called enzymes, are involved in the replication process. These enzymes function to initiate replication at several starting points and ensure fast and efficient replication. Today, the theory of DNA structure and function is clear. The sequence of bases in the DNA of every organism on Earth gives each its unique appearance. The discovery of DNA was one of the greatest scientific achievements of the 20th century.